The overall goal of the following experiment is to analyze the structure-activity relationships of synthesized arthropod neuropeptides on their cognate GPCRs. This is achieved by transfection of Chinese hamster ovary K1 empty cells with the desired GPCR expression vector to obtain stably transformed cell lines derived from a single cell. As a second step, the stable cell line is transfected with the aquarin plasmid, which carries the reporter gene aquarin for measuring intracellular bioluminescence in the calcium bioluminescence plate assay. Next, the calcium bioluminescence plate assay is performed in order to determine cytoplasmic calcium levels upon peptide ligand application to these recombinant cells. Results are obtained that show differences in bioluminescence units upon application of structurally different peptides or different peptide concentrations to the recombinant cells based on elicited intracellular calcium release as detected by the reporter aquarin in the plate assay.
The main advantage of utilizing stably transformed clonal cell lines expressing recombinant GPCRs over transient expression protocols is that receptor expression levels are more uniform, allowing for consistency in results. Further, the targeting of the apoaquarin protein to the mitochondria allows sensitive calcium measurements through bioluminescence. Demonstrating the experiment today will be two graduate students, Shaoling Liu and Simon Kirsch, as well as a postdoc, Suparna Taneja Bageshwar, from my laboratory.
To establish stable cell lines, start by growing Chinese hamster ovary K1 empty cells at 37 degrees Celsius in a 5% carbon dioxide humidified incubator. Maintain the empty cells in growth medium with 1x antibiotic antimycotic. After the cells are growing healthily, seed the Chinese hamster ovary K1 cells into T25 tissue culture flasks. Grow the cells overnight in growth medium without antibiotics until they're about 30% confluent. Prior to transfection, prepare a DNA expression vector containing the GPCR of interest as described in the written protocol. Combine the DNA with 100 microliters of Optimem reduced serum medium. Mix 6 microliters of lipofectin reagent into 100 microliters of F12K serum-free medium and incubate at room temperature for 30 to 45 minutes. Following incubation, gently mix the DNA and lipofectin solutions in a dropwise fashion. Let the mixture stand at room temperature for 10 to 15 minutes. In a 15 ml tube, gently mix the transfection mixture into 1.8 ml of fresh F12K medium in a dropwise fashion. Then, wash the cells with PBS and add this new transfection solution to the cells. Following an 18-hour incubation, change the medium to F12K medium plus 10% FBS without antibiotics and incubate overnight. The following day, split the cells into two T25 flasks with F12K medium plus 10% FBS without antibiotics as described in the written procedure. Incubate for an additional 18 hours. Replace the medium with selective medium. Then culture the cells using the selective medium for 3 to 4 weeks. Over time, this will select for cells that have stably incorporated the plasmid into their genomic DNA. Continue maintaining the cells using maintenance medium. Periodically freeze the cell lines to prevent loss in case of unexpected contamination. To select for clonal cell lines, first trypsinize and centrifuge the cells with maintenance medium as described in the written protocol. Then, resuspend the cells in 5 ml of maintenance medium. Remove 0.5 ml of the cell suspension and add 4.5 ml of fresh maintenance medium to make a 10x dilution and transfer 100 microliters of the 10x dilution into 12 wells of a 96 well plate to select for single cells. Continue to make 10x serial dilutions of these cells about 19 times for a theoretical final suspension of 1 cell per 100 microliters and transfer 100 microliters of each serial dilution into 12 wells of a 96 well plate. After 18 hours of incubation, observe 96 well plates under an inverted light or fluorescence microscope and mark wells that appear to contain only one single cell or two cells that have obviously divided from one single cell. Observe the plates every day, changing the medium every 3 days. Allow the cells to grow for 1 week until about 80% confluent. Then rinse the marked wells with 200 microliters of PBS and trypsinize them with 100 microliters of PBS trypsin EDTA solution. Remove the PBS trypsin EDTA solution, add 100 microliters of maintenance medium, and transfer the cells from each marked well into one well of a 6-well plate with 1 milliliter of maintenance medium. After growing the cells in the 6-well plate for 3 days, transfer the cells into individual T25 flasks. Periodically freeze the cell lines so they can be retrieved if the cells stop performing consistently. To determine the cell line with the highest response, test the transferred cells using the calcium bioluminescence assay with an active ligand such as the receptor peptide ligand or other positive control agonist. The calcium bioluminescence assay will be demonstrated in detail later in this video. Perform the single cell selection for a second time before proceeding with the experiment. When clonal cell lines are chosen for assays and maintained in culture, it is critical to keep track of passage numbers, as high passage number cells may stop performing. In a T25 flask, grow cell lines expressing the desired receptor in maintenance medium to about 90% confluence. After trypsinizing and centrifuging the cells, resuspend them in the maintenance medium. Dilute cells about 10x with maintenance medium and count the cell number with a hemocytometer. Then, adjust the cell number to approximately 2 × 10⁵ cells per milliliter. Seed 2 milliliters of the cells into each well of a 6-well plate and incubate the plate for 24 hours, after which time the cells should be about 60% confluent. Prior to the start of the calcium bioluminescence plate assay, prepare the reporter gene of interest in the expression vector construct. A mitochondria-expressed equorin reporter is used here. Then, transfect the cells with 1 microgram of this reporter plasmid as described in the written protocol. After incubating the cells in a 6-well plate for 24 hours, trypsinize, centrifuge, and resuspend the cells. Count the cell number to 400,000 cells per milliliter and transfer 100 microliters into each well of a 96-well white thin-bottom microtiter plate. Incubate for another 24 hours until about 80% confluence is reached. This is the optimal concentration of cells for the bioluminescence assay. The following steps should be performed in the dark, but are shown here in the light for demonstrative purposes. Prepare enough calcium-free DMEM medium containing 5 micromolar cholinterazine for 90 microliters per well. Remove the old media from the plate and add 90 microliters into each well. Incubate the plates for 3 hours in the dark at 37 degrees Celsius and 5% carbon dioxide, after which the cells are ready to be tested. This demonstration is performed using a Novastar plate reader in bioluminescence mode. To begin, purge the machine pumps. The plate should be placed into the plate holder while in the dark. Solubilize the peptides in a 1.5 ml Eppendorf tube using enough calcium-free DMEM media for 10 microliters of 10x peptide per well. Set the aspirate depth and position determination of the peptide solution before use. Challenge the cells with 10 microliters of the peptide and immediately begin recording the light emission. In this demonstration, the instrument is set to record the light emission at 465 nanometers for each well every 2 seconds for a total time of 50 seconds. After the run is complete, wash the machine pumps. Repeat the process for each peptide sample. Save the data and wash the machine pumps again. Following the assay, the data of light emission from each well is automatically transferred into a Microsoft Excel datasheet by the software program. Paste the data from Excel to Prism Software 4.0. Generate a graph with the various peptide concentrations in the x-axis and bioluminescence units in the y-axis. Select a nonlinear regression curve fit analysis to obtain concentration response curves for each peptide and their corresponding EC50 values. Each experiment should be repeated 3 times for data analysis. When expressed in Chinese hamster ovary K1 cells, the kinine receptor from the mosquito Aedes aegypti behaved as a multi-ligand receptor and functionally responded to concentrations as low as 1 nanomolar for the 3 endogenous Aedes kinines tested singly using the calcium bioluminescence plate assay. Statistically different EC50 values of about 16 nanomolar for Aedes aegypti kinine 3, 26 nanomolar for Aedes aegypti kinine 2, and 49 nanomolar for Aedes aegypti kinine 1 were determined, resulting in a rank order of potency of Aedes kinine 3 as the most potent and Aedes kinine 1 as the least. Shown here is the activity comparison of 6 alpha-amino isobutyric acid kinine analogs on the tick kinine receptor expressing Chinese hamster ovary K1 cell line by a calcium bioluminescence plate assay. The y-axis represents percent maximal bioluminescence units for each analog expressed as a percentage of bioluminescence observed at a concentration versus the maximal response observed at all concentrations tested for each analog. Analog kinine hexapeptide FFF-SWG amide is the positive control for receptor activity. the single replacement kinine analog F aminoisobutyric acid WG amide was more active than the positive control hexapeptide kinine analog. The kinine analog with two aminoisobutyric acid replacements, aminoisobutyric acid FF aminoisobutyric acid WG amide, was the most potent of the double replacement analogs tested.
After watching this video, you should have a good understanding on how to express a GPCR and obtain clonal cell lines to analyze structure-activity relationships of ligands on receptors by a calcium bioluminescence assay.